In this video, we will show you how to calculate MAR and RAL limits. You need to calculate these limits as the acceptance criteria during cleaning validation of therapeutic products. MAR sets the allowable residue, and RAL converts those limits into swab and rinse acceptance criteria. Why do residue limits matter? Residue limits help prevent carryover from one product to the next. They protect product quality and patient safety. They also make cleaning validation measurable and documented. In a typical changeover scenario, product A is made first. The equipment is then cleaned and checked. Product B is made next on the same equipment. The residue from product A must remain within acceptable limits. The two key terms used in the residue limit calculation are 1. MAR, which stands for Maximum Allowable Residue. It is the maximum residue allowed to carry over from the previous product into the next product. And 2. RAL, which stands for Residue Acceptability Limit. It is the practical testing limit for swab or rinse samples. In this video, we will cover the step-by-step calculation of MAR and RAL. First, we will gather the inputs and select the safety factors. Then we will calculate dose MAR and toxicity MAR. Finally, we will calculate the swab and rinse RAL acceptance limits. By the end, you will know how to calculate cleaning limits logically and consistently for the cleaning of therapeutic products in pharmaceutical manufacturing. Now, let us begin with Step 1. Step 1 is to gather the required inputs. To simplify, let's look into all the inputs first. T is the minimum therapeutic dose of the previous product. B is the maximum daily dose of the next product, expressed as units per day. C is the unit weight of the next product. L is the smallest batch size of the next product. EW is the shared product contact surface area. AS is the swabbed area. WR is the rinse quantity. If toxicity is assessed, we also need toxicity data such as LD50, NOEL, or ADI. Good inputs are essential, as incorrect inputs will yield an incorrect residue limit. Step 2 is to select the safety factor and the default limit. For this oral solid dose example, we will use a safety factor of 0.01. The default therapeutic product MAR is 10 parts per million. After calculating dose MAR and toxicity MAR, you have to compare both values with 10 parts per million. You should select the lowest value as your swab and rinse acceptance limits. The lowest value gives the most stringent acceptance criterion. Step 3 is to calculate dose MAR. The formula is MAR-T equals T multiplied by 10 to the power of 6, multiplied by the safety factor, divided by B multiplied by C. The result is expressed as milligrams of previous product residue per kilogram of the next product. This is also expressed as parts per million. A lower therapeutic dose or a lower daily intake of the next product usually results in a stricter MAR. Step 4 is to calculate toxicity MAR. First, calculate NOEL from LD50. NOEL stands for No Observed Effect Level. It means the dose level at which no harmful or pharmacological effect is observed. NOEL equals LD50 multiplied by 0.0005 multiplied by 70. Then calculate ADI. ADI stands for Acceptable Daily Intake. It means the amount of a substance that a person can be exposed to each day without expected harmful effects. ADI equals NOEL multiplied by 0.01. Then calculate toxicity MAR. Toxicity MAR equals ADI multiplied by 10 to the power of 6, divided by B multiplied by C. Finally, compare the dose MAR, toxicity MAR, and the default 10 parts per million limit. Use the lowest value. Now, let us apply the calculation in practice. You are watching an example as a training. Say, the previous product you made was cetirizine hydrochloride, 10 mg tablets. The next product will be ibuprofen, 200 mg tablets. For this example, the minimum therapeutic dose, T, is 10 mg. The maximum daily dose of the next product, B, is 5 tablets per day. The tablet unit weight, C, is 300 mg per tablet. So, B multiplied by C equals 1,500 mg per day. The batch size, L, is 500 kilograms. The shared equipment surface area, EW, is 320,000 square centimeters. The swab area, AS, is 100 square centimeters. The rinse quantity, WR, is 400 kilograms. The LD50 used for the example is 400 milligrams per kilogram. Now calculate the dose MAR. MART equals T times 10 to the power of 6 times safety factor, divided by B times C. Substitute the respective values. MART equals 10 multiplied by 10 to the power of 6 multiplied by 0.01 divided by 5 multiplied by 300. This equals 66.7 milligrams per kilogram. Therefore, the dose MAR equals 66.7 parts per million. Now calculate toxicity MAR. NOEL equals 400 multiplied by 0.0005 multiplied by 70. This equals 14 milligrams per day. ADI equals 14 multiplied by 0.01. This equals 0.14 milligrams per day. Toxicity MAR equals 0.14 multiplied by 10 to the power of 6 divided by 5 multiplied by 300. This equals 93.3 milligrams per kilogram. So, the toxicity MAR equals 93.3 parts per million. Now compare all three values. Dose MAR is 66.7 parts per million. Toxicity MAR is 93.3 parts per million. The default MAR is 10 parts per million. The lowest value is 10 parts per million, so the selected MAR is 10 parts per million. Now use the selected MAR to calculate the swab limit. RALT for swab equals MAR multiplied by L multiplied by AS multiplied by 10 to the power of 3 divided by EW. Substitute the respective values. RALT equals 10 multiplied by 500 multiplied by 100 multiplied by 10 to the power of 3 divided by 320,000. This equals 1,562.5 micrograms per swab. On a surface basis, this is 15.63 micrograms per square centimeter. Therefore, the swab acceptance limit is 1,562.5 micrograms per swab. Now calculate the rinse, RAL, and final acceptance limit. RALT for rinse equals MAR multiplied by L divided by WR. Substitute the respective values. RALT equals 10 multiplied by 500 divided by 400. This equals 12.5 milligrams per kilogram of rinsate. That is equivalent to 12.5 parts per million. So the final swab limit is 1,562.5 micrograms per swab. The final rinse limit is 12.5 parts per million. These are the cleaning validation acceptance limits for this therapeutic product changeover example. If you have any questions or would like to learn more about this topic, please visit our website. You can find in-depth information on cleaning validation protocols in the pharmaceutical industry in our blog. Please see the article link in the description below. Subscribe with us to gain access to a library of hundreds of well-written GMP documents. Use the link in the description to reach us. Thanks for watching.